I am Dr. Cecily Assistant Professor of Zoology, Holy Cross College, Tiruchirappalli. And today I am going to discuss about the glycogen metabolism. And welcome back to the biochemistry class. So the learning objectives of today's session. So after studying this module, the students will be able to describe the pathway of glycogen synthesis, that is glycogenesis, in liver and muscle tissue, and describe the pathway of glycogenolysis and what are the key enzymes of this pathway and how this pathway is regulated and what is the role of hormones in the regulation of this pathway. Now we will move on to glycogenesis, that is the synthesis of glycogen from glucose. So you all know glycogen is nothing but it is a storage form of carbohydrate. So it is a form in which carbohydrate is stored as glycogen in the liver tissue as well as in the muscle tissue. And which part of the cell it is stored? It is stored in the cytosol as glycogen granules. When actually the glycogenesis takes place, this takes place when the blood glucose level is high. So after the fed condition, after a heavy breakfast or lunch, etc., when the glucose level rises in the blood, insulin comes into action and stores all the excess glucose in the form of glycogen in the liver and the muscle tissue. And what is the function of the glycogen in the liver? To maintain the blood glucose level. So whenever glucose level falls, the glycogen breaks down in the liver, comes out of the liver cells and bring back the glucose to the normal level. In the same way, whenever glucose level increases in the blood, glycogen liver cells takes up all the excess glucose and convert it to glycogen. And what is the need to maintain the blood glucose level? Because for example, brain cells cannot live even for 5 minutes, not more than 5 minutes without glucose. So it is blood glucose level should be meticulously maintained. And what is the main function of glycogen that is stored in the muscle? The main function is to give energy or fuel to the tissue, to the muscle tissue. And this slide shows a glycogen molecule. As we see here, it has thousands and thousands of glucose molecule. And how these glucose molecules are arranged, a small portion of this glycogen is shown here. It's here enlarged. You can see hundreds of glucose molecule. They are attached by alpha-1,4 bond. And at branch point, the linkage is alpha-1,6. So we will see the steps that is involved in glycogenesis. What is the very first step? The very first step is synthesis of glucose 6-phosphate from glucose. So which enzyme catalyzes the reaction? HK and GK. Hexokinase is present in muscle tissue. It is present almost virtually in all the tissues in our body, whereas glucokinase is present mainly in the liver cells. And here this enzyme GK is inducible by insulin, whereas the enzyme HK is not inducible. So HK will be active even in fed condition and starvation condition, and GK will come into action only in fed condition. And this conversion of glucose to glucose 6-phosphate is the same first step in glycolysis process also, as you have studied. Step 2, G6P is converted to G1P by a simple enzyme phosphoglucosamidase. It is a reversible reaction. So glucose 6-phosphate is converted to glucose 1-phosphate. Now the third step is the step in which the active form of glucose, the form in which glucose is incorporated into the glycogen molecule, that is UDP glucose, is synthesized. So here the glucose 1-phosphate is activated by complexing with UTP. So from UTP, UMP is transferred to glucose 1-phosphate and UDP glucose is synthesized. Plus inorganic pyrophosphate is liberated, and this inorganic pyrophosphate is acted upon by the enzyme pyrophosphatase, and the hydrolysis of this PPI gives the energy for the synthesis of UDP glucose. And UDP glucose is activated form of glucose, the form in which it is incorporated into the glycogen. And coming to step 4. So in step 4, the UDP glucose is ready. The carbon 1 of the UDP glucose, the glucose that present in the UDP glucose, the carbon 1 attaches to the hydroxyl group in the carbon 4 of the terminal glucose list. So we have a glycogen molecule with so many branches, and each branch has a terminal glucose molecule, and this UDP glucose is the activated form of glucose which can get attached to the 4th carbon of the terminal glucose. So what is the bond that is synthesized? It is alpha-1,4 bond, and that process is done by glycogen synthase. So this job is done by the enzyme glycogen synthase, which is the key enzyme, the rate limiting enzyme for this pathway. And glycogen synthase catalyzes the formation of alpha-1,4 glycosidic bond. So for this reaction to occur, already an existing glycogen primer is needed. Only when glycogen primer is present, the glycogen synthase can add the activated glucose to the primer molecule. So what is a primer? Primer is nothing but a small protein molecule about 37 kilodalton, which is called by the term glycogen, and to this glycogen a glucose residue is attached to a specific tyrosine residue, and again glycogen also catalyzes the attachment of another 7 to 8 glucose molecules to the glycogen. So this 7 to 8 glucose molecule attached to the protein glycogen is what we call by the term glycogen primer. Once glycogen primer is ready and activated glucose is ready, glycogen synthase can go on add glucose to the glycogen. So we get a big glycogen molecule with thousands and thousands of glucose. Okay. So next we will move to the step 5. So again the most important thing is for glycogen synthase, how many glucose can be added in a branch? Not many glucose molecule. So a maximum of about only 13 glucose molecule can be added. The next glucose cannot be added by glycogen synthase. So step 5, branching enzyme comes into action. So what is the job of the branching enzyme? To carry this 6 glucose residue. Okay, the alpha 1,4 chain has about 13 glucose residue, about 6, a minimum of 6 glucose residue is transferred by the branching enzyme, and that transfers it to a new branch point, and this is an energy requiring process which requires 1 ATP and 1 ETP, 1 ATP and UTP. Here you can see the illustration shows a branch of glycogen tree, and what is the job of glycogen synthase? So here to differentiate we have added C14 labeled glucose. So the mustard color glucose is already which is present in the glycogen primer. C14 glucose is newly added. The glycogen synthase enzyme adds a glucose. So we can see here glucose is added and about chain length grows to about 11 to 13 glucose residue. So after 13 glucose residue, the glycogen synthase can no more add glucose molecule. Which enzymes comes into action? The branching enzyme comes into action. You can see about 6 glucose residues are taken from one branch and then it is added to the neighboring chain at alpha 1,6. Already whatever glucose molecule is present, it is in alpha 1,4 linkage. So the first and the fourth carbon is in linkage. So this branch is attached to the 6th carbon, establishing a new branch point, that is alpha 1,6. So now we have come to the end. So this is a conclusion of the glycogenesis process. Glucose is converted to glucose 6-phosphate by HK or GK. G60 is converted to glucose 1-phosphate by phosphoglucomutase, and glucose 1-phosphate is converted to UDP glucose catalyzed by the enzyme. Two enzymes are there, one is the uridyl transferase, the other one is the enzyme is a pyrophosphatase, and alpha 1,6 glucosylate unit together is called by the term glycogen. So we have seen the 5 steps that are involved in glycogenesis, a little bit about the regulation of glycogenesis. Here, as we know, the process of glycogen synthesis is a storage form of carbohydrate. So when glucose will be converted to glycogen? Only in fed condition when the blood glucose level increases. So which hormone will come into action? Insulin. So what is the function of insulin? Insulin is a storage hormone. So whatever excess glucose, it stores it in the glycogen in the liver tissues as well as in the muscle tissue. So that is the insulin and glucagon are the 2 hormones that keeps the blood sugar homeostasis. Insulin decreases blood glucose level, whereas glucagon increases the blood glucose level. So this enzyme is present either in the active or inactive form. It is present in the inactive form in the dephosphorylated form. Inactive form is the phosphorylated form. So addition of phosphate to the enzyme or removal of phosphate to the enzyme is done by cyclic AMP and phosphatase enzyme. So cyclic AMP by cyclic AMP dependent protein kinase adds phosphate to glycogen synthase and makes it inactive. And cyclic AMP level is increased by glucagon, whereas cyclic AMP level is decreased by insulin. So this is a small, you can see here epinephrine and glucagon are the 2 hormone that increase, that will come into the blood when the blood glucose level rises. These 2 hormone increases cyclic AMP level, which in turn increases cyclic AMP dependent protein kinase, and that in turn will add a phosphate group to glycogen synthase and makes it inactive. So where from the phosphate group comes, ATP is converted to ADP. So we have completed one, that is glycogenesis, synthesis of glycogen. So we will quickly go through the breakdown of glycogen, a process called glycogenolysis. So this again involves about 4 steps. The very first step in glycogen phosphorylase is removing. So we have a glycogen molecule with thousands of glucose molecule. So this enzyme glycogen phosphorylase will remove the terminal, so outer branches. So from the outer branches, all the terminal glucose residues, or it is called by the term phosphorolytic cleavage. That is, they go and add a phosphate group and cleaves glucose as glucose 1-phosphate. So what will happen? The next one will become the terminal glucose. So that is again removed by phosphorylase. So phosphorylase will go on removing glucose from all the outer branches until about, so the process goes on until about 4 glucose residues are left on either. So if there are 13 glucose molecule in a branch, the 13, 12, 11, 10, everything will be removed by phosphorylated cleavage, catalyzed by the enzyme glycogen phosphorylase, the key enzyme, the rate limiting enzyme of this pathway, until about 4 glucose residues are left on either side. So this glucose molecule, so we can see in the illustration here, you can see the phosphorylase removing all glucose molecule. You can see in the next only 4 glucose residues are left on either side of the branch point. Which enzyme will come into action? The enzyme called glucan transferase. So what this glucan transferase does? It takes 3 glucose residue from there are 4 left on either side, isn't it? So 3 glucose residues are transferred from one branch to another branch so that the alpha-1,6 linkage is exposed. Can you see here? So branch point is exposed. So what will happen? The third enzyme, debranching enzyme, comes and cleaves the alpha-1,6. So this enzyme is called alpha-1,6 glucosidase. So debranching enzyme is alpha-1,6 glucosidase, cleaves the alpha-1,6 linkage. Now we have a straight chain with all alpha-1,4 bond, and again phosphorylase comes into action, and that will cleave all glucose as glucose 1-phosphate. So in step 4, G1P, all G1P which we get from breakdown of glycogen is converted to glucose 6-phosphate in both the liver cells as well as in the muscle cells. What happens here is G6P in the cell is trapped. Why we say it is trapped? Because we don't have a transport system for G6P. So when there is no transport system for G6P, G6P cannot come out of the cell. So G6P must be converted to glucose by the enzyme glucose 6-phosphatase if it needs to come out of the cell. And this enzyme glucose 6-phosphatase is not present in all cells, it is present only in the liver cells and to a certain extent in the intestine and kidney. So liver only plays a major role in converting glucose 6-phosphate to glucose, and all this glucose will come out. Thousands and thousands of glucose molecule will come out of the liver cells and it will bring back the blood glucose level to the normal level. And what happens to the G6P in the muscle? They enter the glycolysis and that gives the fuel energy or ATP to that particular cells. So we have seen both the steps involved in glycogen synthesis and the steps involved in glycogen breakdown. And coming to the glycogen phosphorylase enzyme, we saw in glycogen synthase enzyme, it is active in the dephosphorylated form, inactive in the phosphorylated form. Coming to glycogen phosphorylase, it is just the opposite. Here it is active in the phosphorylated form and it is inactive in the dephosphorylated form. And we need to remember, whenever glycogen synthesis is taking place, breakdown must be inhibited, and whenever breakdown is taking place, synthesis must be inhibited. It's like a person is constructing a wall and another person is just demolishing the wall. What will be the net result? There will be no wall. So whenever synthesis is taking place, breakdown should be inhibited, and when breakdown is taking place, synthesis should be inhibited. And that job is done by, for example, if you take phosphorylase, it is kept in active form by the enzyme phosphorylase kinase that adds a phosphate group to glycogen phosphorylase, makes it active. And this phosphorylase kinase is again under control of cyclic AMP. Cyclic AMP dependent protein kinase will add phosphate to phosphorylase kinase, and cyclic AMP dependent protein kinase is under the control of the hormone glucagon and epinephrine. So glycogen phosphorylase is converted to the active form by cyclic AMP. It is converted to the inactive form by the enzyme phosphatase. When phosphate is removed by phosphatase, and that job is done by insulin. So these are the 3 hormones, and another method is by allosteric. Whenever energy status of the tissue is high, what will happen? Breakdown will not occur. When high AMP is there, that is tissue energy status is less, breakdown will take place. When the high ATP, that is energy status of the tissue is high, breakdown will not take place, instead synthesis will take place. And all this job is done by 3 hormone, glucagon, epinephrine and insulin. They will dictate whether glycogen synthesis must take place or glycogen breakdown must take place. So for example, insulin will increases the uptake of glucose because this is an anabolic hormone. It comes into action in a fed condition when your blood glucose level increases. So insulin increases glucose uptake by the cell. It promotes glycogen synthesis from the excess glucose, increases glucokinase activity, keeps glycogen synthase and glycogen phosphorylase in the dephosphorylated form. So it actually promotes phosphatase activity. By promoting phosphatase activity, what it actually does? It removes the glucose. So here the collective, you can see here glycogen being converted to glucose 1-phosphate and glucose 1-phosphate to glycogen. So here glycogen synthase is a key enzyme in synthesis, phosphorylase is a key enzyme in breakdown. So what is the job of insulin? Promote the activity of protein phosphatase. So what will happen? Phosphate is removed, synthase is active. The same way they protein phosphatase, actually phosphate is removed, phosphorylase becomes inactive. What is the job of glucagon and epinephrine? Increases intracellular cyclic AMP level, cyclic AMP dependent protein kinase is activated, adds phosphate to glycogen synthase, makes it inactive. Here adds phosphate to glycogen phosphorylase, makes it active. And please remember, when glucose level, when we have taken a heavy meal, when the glucose level is increased, there is no need for glycogen to be broken down in the liver. So again, so much of glucose coming out of the liver will further increase the glucose level. So under fed condition, the glycogenolysis must be inhibited, glycogenesis must be promoted. It's very simple. And in case of when in starvation, like we have not taken food for past 13 hours, 15 hours, glucagon and epinephrine will come into action and they will promote the breakdown and they will inhibit the glycogen synthesis. So to summarize what we have discussed, first thing, the glycogen is nothing but it is the storage form of carbohydrate in the mammalian liver and muscle cells. These are the most important tissues where glycogen is stored, and it is stored about 6% in the liver cells and about 0.7% in the muscle cells. And main function of the glycogen in the liver cells is to maintain the blood glucose level. Whenever glucose level comes down or goes up, either glycogen will be broken down or glycogen will be synthesized. And what is the main function of glycogen in the muscle? To provide energy or fuel to the cell when the muscles are active. And which is regulating? One is cyclic AMP dependent protein kinase. Actually they phosphorylate glycogen synthase, makes it inactive, and they phosphorylate glycogen phosphorylase and makes it active. And that is promoted by the two hormone epinephrine and glucagon. And insulin acts reciprocally. So the action of insulin is just the opposite to the glucagon and epinephrine. It actually inhibits glycogenolysis, as we all know, and we have seen how it inhibits glycogenolysis and how it promotes glycogenesis. It promotes glycogenesis by a simple process, that is, it actually removes phosphate. So again, it removes phosphate from glycogen phosphorylase. So removal of phosphate from glycogen phosphorylase makes the enzyme inactive. So that is a job done by insulin. And these are few questions. You can just go through the questions and answer. So these are some of the questions, and thank you very much. These are the books for references, and thank you very much for the patient listening. Thank you.